Okay. Good afternoon, everybody. It's a pleasure to have once again another session of the immuno-webinars that is a joint program from the Institute of Biophysics at the Federal University of Rio de Janeiro and the Institute of Biological Sciences at Universidade de São Paulo. And today we have the honor to host Dr. Eugenio Hottz, who got his bachelor's degree in biology in 2008 at the Centro Universitário Barra Mansa in Rio, master's degree in tropical medicine in 2010, and got his PhD in cellular and molecular biology in 2014 from the Oswaldo Cruz Institute, Fiocruz, under the supervision of Drs. Patricia Boza and Fernando Boza, with a sandwich period at University of Utah through the PDSC campus program in Guy Zimmerman's group. He was also a postdoctoral fellow at the Immunopharmacology Laboratory at the Instituto Oswaldo Cruz, and a recipient of Nota 10 postdoctoral fellowship from FAPENG. He's currently an assistant professor at the Biochemistry Department of the Federal University of Juiz de Fora, and in the postgraduate program in biological science. Eugenio has received several awards. He's a 1D Fellow of Research Productivity Fellowship from CNPq, and among national and international grants, he's a 2022 grant recipient of Instituto Serra Pileira. He has published 53 papers and concludes the supervision of 6 master's students and 2 PhD students. His lines of research include investigating the mechanism of activation of the innate and adaptive immune response during infection by dengue virus, HIV, and SARS-CoV-2. He works mainly on issues aimed at the understanding the participation of platelets in the pathophysiological mechanisms of viral infections, mainly regarding immune, inflammatory, and thromboembolic mechanisms, induced in leukocytes and endothelial cells through interaction with activated platelets. His work also includes the investigation of metabolic changes in viral infections and mitochondrial involvement in the process of immune activation of cell death. So it's a great pleasure to have you here today with us, Eugenio, and the floor is yours.
Thank you, Bruno, for the introduction. So I'll talk about thromboinflammation today. So thromboinflammation is this intersection area between coagulation and immunity. So we are very interested in those cellular and molecular events originally described to the coagulation, but that regulate innate and adaptive immune responses. Most of the work done by our group in this field was on viral infections, and thromboinflammation is very present in viral infection, including that obvious contributions to the pathogenesis as microvascular thrombosis, thrombocytopenia, hemorrhage, and thromboembolic events that obviously contribute to multi-organ dysfunction, but also by regulating inflammatory infiltrate, the cell-mediated cytotoxicity, and systemic inflammation that also contributes to shock and hemodynamic dysfunction and multi-organ impairment, leading to cases of viral sepsis. Recently, we have all noticed that patients with COVID-19 are at higher risk of thromboembolic events, especially pulmonary and embolism and deep vein thrombosis. And this is a post-mortem histopathological investigation of lungs of patients from COVID-19, and they show these pulmonary microvascular thrombosis leading to partial or complete obstruction of the pulmonary arterioles. And these in COVID-19 were 10 times more frequent than in deaths by influenza pneumonia. So to talk about thromboinflammation, I need to talk about platelets. Platelets circulating in a resting state and become activated when getting contact with proteins from the extracellular matrix. or when activated by agonist generation by coagulation activation. So activated platelets become morphologically change, and the activation of the intergreen alpha 2b beta 3 is an important event because this is the fibrinogen receptor in platelets, leading to platelet aggregation and formation of the platelet clot with the fibrin mesh. The main pathway for the activation of coagulation is the extrinsic pathway or tissue factor pathway. The name tissue factor or extrinsic pathway is because this protein is constitutively expressed by the cells outside the vessel. So when we have a vessel rupture, the coagulation factors in plasma get in contact to tissue factor that initiate the coagulation cascade. However, it was recently shown that intravascular cells that do not express tissue factor constitutively can present regulated expression of tissue factor under inflammatory or infectious stimuli. So monocytes, neutrophils, endothelial cells, and even the platelets themselves can express tissue factor in the intravascular space, contributing to the higher risks of intravascular coagulation. Another component that links the inflammation with coagulation are platelets themselves. Activated platelets perform a lot of response that participate in the immune regulation. Platelets have stored granules with a lot of chemokines and cytokines and growth factors. Some of those are exclusively expressed by platelets as platelet factor 4 and platelet-derived growth factor. Platelets also synthesize eicosanoids from the arachidonic acid metabolism. Some of those are also exclusive of platelets. And despite the absence of nucleus, platelets can can produce newly synthesized proteins because they have stored pro-mRNA molecules containing the introns, and depending on stimuli, platelets can remove those introns of selected mRNA molecules, being able to regulate the expression of new proteins. Some examples are IL-1 beta and the tissue factor, which is expressed exclusively by activated platelets. Besides, platelets has a lot of pattern recognition receptors, being able to recognize and respond to pathogens. Most of the toll-like receptors, for examples, are expressed by platelets, by some of the cytosolic sensors and also cytokines and chemokine receptors. This way, platelets can act as sensors to infection. And of course, many viruses can usurp these receptors to enter the platelets, and the platelets can also recognize viral pumps and initiate response to this virus, including production and secretion of cytokines, newly synthesized or stored in granules, but also in activation of pathogens by their products. Platelets can also interact with a lot of immune cells. This protein, which is expressed in the membrane of granules, is called p-selectin, is translocated to platelet surface under the activation stimuli, and is the main molecule to the interaction of platelets with leukocytes. And besides p-selectin, the integrin alpha 2b beta 3 can connect platelets with leukocytes through fibrinogen bridging the platelet and the leukocyte fibrinogen receptors. And of course, these adhesion molecules interaction signals to the leukocytes. Therefore, platelet-leukocyte interaction regulates a lot of important leukocyte responses, as net extrusion by neutrophils, activation of inflammatory genes as COX-2 and cytokines by monocytes and by lymphocytes. And finally, platelets have been also described to act in the extravascular space. So it has been shown that the infiltration of platelets to inflammatory sites as the synovia of rheumatoid arthritis patients, or the lung in patients with influenza pneumonia. In influenza pneumonia, the infiltration of platelets and activation of platelets in the vasculature can contribute to major pathological events as endothelial activation and increased plasma leakage, contributing to pneumonia, but also to the recruitment of neutrophils that are essential to the viral clearance. So our hypothesis is that platelet activation in the intravascular or the extravascular milieu contribute to pathological or protective responses during viral infections. Most of our work were done in COVID-19 and dengue, but we have some work in influenza, HIV as well, chikungunya virus, either. And this platelet activation can contribute to major immunopathological events as leading to hyperinflammation, hypercoagulability, severity, and mortality in severe viral infections and viral sepsis. So most of my work was performed in dengue. We have shown that our group and others have shown that in parallel to the reduction of platelet counts during dengue, a lot of platelet-derived vasoprotective factors, as angiotensin, sphingosine 1-phosphate, thromboxane 2, are also reduced. But at the same time, the platelet is still in circulation and activated and produce a lot of vasoactive molecules and pro-inflammatory molecules, contributing to the increased vascular permeability in dengue. So we have shown increased platelet activation in patients with dengue, which was higher in patients with more severe clinical presentation. And besides that, platelets also interact with monocytes during the infection, regulating the production of pro-inflammatory molecules, which was also associated with increased vascular permeability. In this field of research in our group, we have reported, this was a publication from my PhD, we have reported that platelets from patients with dengue or platelets infected with dengue virus in vitro induce the production of IL-1 beta, and we have shown that platelets have all the components for the NLRP3 inflammasome, which is functional and process the newly synthesized IL-1 beta to be secreted in extracellular vesicles, and those platelet extracellular vesicles can activate endothelial cells through the L1 receptor, increasing endothelial permeability in vitro. And these responses of production of IL-1 beta containing microparticles, activation of inflammasome, and synthesis of IL-1 beta were all associated with increased clinical signs of increased vascular permeability in patients with dengue. We have also shown the increased formation of platelet monocyte aggregations. So here we have the CD14 positive monocytes, and we inquired for the expression of the platelet marker CD41. And these double positive population are monocytes that are decorated with platelets on their surface, and it's increased in dengue patients, correlate with clinical signs of increased vascular permeability, and we have shown that these interactions with platelets induce the production of pro-inflammatory cytokines in monocytes. Well, when we talk about platelets and viruses, it's natural to ask if the virus can infect the platelets. The interaction of a virus with a host cell initiated by the interaction with a surface receptor that mediates the internalization of the virus. And the viruses, they have mechanisms to escape this endosome before they merge with the lysosomes, releasing their viral genome to the cytoplasm. In the case of dengue virus, for example, which is a positive sense viral RNA, it's read as a messenger RNA by the cell, leading to the expression of the viral proteins. So the viral enzymes, together with the cell machinery, can assemble new viral particles and release new viruses. So are platelets able to translate viral RNA and replicate viruses? We investigated this for dengue virus. So we infected platelets with the virus and investigated the expression of a non-structural protein, NS1. The non-structural proteins are not part of the viral particle, but they are expressed by the translation of the viral genome. So when we see accumulation of NS1 in platelets infected by dengue virus, in these assays, we have used purified virus to make sure to not have contaminating NS1 from other cells. So platelets were able to translate the viral RNA and to replicate the viral RNA as we detected the accumulation of viral RNA in the platelet pellets, but not in the supernatant, indicating that platelets were not able to assemble a new viral particle and release them. However, we observed the secretion of NS1 itself to the extracellular milieu. Why is this important? It's important because NS1 has been recognized as a viral pump, able to activate leukocytes and endothelial cells through toll-like receptor 4. So we then asked, are there roles for NS1 in platelet activation by dengue virus? We have shown that the NS1 secreted by infected platelets or exogenous NS1 were both able to activate toll-like receptor 4 in platelets. And this is important for platelet activation by dengue virus in vitro, since when we infect platelets in the presence of anti-toll-like receptor 4 antibodies or anti-NS1 antibodies, we partially reduced platelet activation by dengue virus infection. So what about SARS-CoV-2? We have also collected evidence of the ability of platelets to translate and replicate SARS-CoV-2 genome. This is not from our group, but this research showed the presence of the positive sense and the negative sense SARS-CoV-2 RNA in platelets from patients. The presence of the negative sense RNA itself is already an evidence of viral replication. And in our group, we were able to recover the whole sequence of the SARS-CoV-2 RNA in platelets from patients with SARS-CoV-2, confirming that we have the complete viral genome in platelets. However, when we get the supernatant of the platelets from patients or in vitro infected platelets input and put these in replicative competent cells, we do not detect infective viral particles from platelets. So again, viral replication in platelets is a non-productive replication cycle. But we were able to investigate the mechanisms that SARS-CoV-2 infection leads to platelet activation. This is a phosphoprotein array when we investigate 48 phosphorylation sites in platelets from healthy volunteers or COVID-19 patients. And I would like to highlight many proteins from the mTOR pathway, which were phosphorylated in platelets from COVID-19 patients. And when we infect platelets in vitro, they become activated. This is an adhesion assay when we put platelets in fibrinogen-coated surface, and activated platelets adhere and spread to a higher area. But when we infect platelets in the presence of the mTOR pathway inhibitor rapamycin, we have a partial reduction of this platelet adhesion to fibrinogen. We investigated a lot of other platelet responses, which were also dependent on mTOR in platelets infected with SARS-CoV-2. And we showed that platelets become activated under infection to SARS-CoV-2 and initiate many responses that were dependent on the mTOR pathway, as secretion of mediators, expression of adhesion molecules, and adhesion and spreading to the fibrinogen, but also some response that were mTOR independent. So, changing a little bit to patients, we have done a lot of work in 2020 during the pandemic to study the pathogenesis of COVID-19, and we provided evidence of platelet activation and hyperactivity during COVID-19, and platelet leukocyte interactions that platelet leukocyte interactions regulating important leukocyte functions, contributing to hypercoagulability and amplification of inflammation during COVID-19. I will show some of this data. Platelets isolated from peripheral blood from patients with COVID-19 shows increased signs of activation, including the expression of P-selectin and CD63. But different from dengue, we have observed that exclusively in the severe COVID-19 patients. We didn't observe increased platelet activation in patients with mild COVID-19. And we also have access to the tracheal aspirate of those patients with severe COVID-19 that were under mechanical ventilation. And we inquired for platelet-derived mediators. Here we quantified those that are exclusive from platelets. And these show that at least the products of platelet activation can get to the airways during severe COVID-19. We don't have strong evidence that the platelets themselves are there. And importantly, both the levels of platelet products in the airways and platelet activation in the blood were strongly associated with poor outcomes during COVID-19. So we had those samples in the admission when platelets were admitted to the ICU, and these patients were followed up to 28 days by a clinical research team, and increased platelet activation and increased monocyte tissue factor expression in the admission were strongly predictive of death. So we investigated whether platelet-leukocyte interactions would have a role in monocyte activation and increased tissue factor expression in those monocytes. Here we show in confocal fluorescence microscopy of whole blood that leukocytes, mononuclear leukocytes, are circulating aggregated with platelets in these patients with severe COVID-19, but not with mild COVID-19. Looking a little bit to the lymphocytes, we observe increased platelet lymphocyte aggregate formation in both CD3, CD4, and CD8 lymphocytes. And in the CD8 positive lymphocytes, we have an interesting data that the lymphocyte that is circulating aggregated with platelets are the ones exerting cytotoxic activity, evidenced here by the CD107, which is a marker of a cytotoxic granule. And in the monocyte, we also show increased platelet monocyte aggregate formation, and those monocytes that circulate aggregated with platelets are the ones expressing tissue factor. The monocytes that circulate freely express significantly less tissue factor than the monocytes that circulate aggregated with platelets. So at this point, we don't know whether the procoagulant and activated monocytes are more likely to form aggregates with platelets, or whether the activated platelet aggregate with monocytes and then stimulate them to become activated and to express tissue factor. So to answer this question, we performed an ex vivo platelet monocyte interaction experiment in which we isolate platelets and monocytes from healthy volunteers and from COVID-19 patients, and exposed control monocytes to platelets from severe COVID-19 patients or from a different person, from a different healthy volunteer. And we show that platelets from severe COVID-19 patients form higher aggregates with monocytes and induce higher expression of tissue factor in a naive monocyte. Those platelets from COVID-19 patients also induce CD16 expression, a marker of inflammatory monocytes. And however, these were at a later time point, at a later kinetics. We observed CD16 expression at 18 hours post-interaction, and tissue factor was a fast response. We observed tissue factor expression at 2 hours post-interaction. We also investigated whether the monocyte that come from a COVID-19 patients is already more likely to interact with platelets. So we incubated those monocytes from COVID-19 patients with platelets from healthy volunteers. Observe that these monocytes from COVID-19 patients, they already has more platelets adhere on their surface, but they recruit more platelets, and increase their tissue factor expression. So both phenomena are likely to happen. We have monocytes more likely to interact with platelets, but we also have platelets more likely to interact with monocytes. And we also investigated if those platelets regulate the production of inflammatory mediators in monocytes. So we performed a multiplex cytokine assay. Of course, many mediators are induced by the interaction with any platelets, regardless the patient or healthy volunteer source. But we found a cluster of mediators that were exclusively induced by platelets from COVID-19 patients. And we were very interested in these mediators, which were TNF-alpha, IL-1 and IL-8, very pro-inflammatory mediators. So before investigating the mechanisms involved in platelet induction of these mediators in monocytes, we confirmed this data in our in vitro model. So we infected platelets, monocytes, and platelet monocytes co-cultures. And we confirmed that platelets are able to respond to the pathogen, increasing platelet activation and the secretion of many mediators. The monocytes were also able to respond to the viral infection, increasing the secretion of many mediators. But when we infected the monocyte in the presence of platelets, we found a higher diversity of mediators secreted by the monocyte in response to the infection. And the monocyte also secreted these mediators in higher levels than monocytes infected alone. Importantly, this interaction of platelets and monocytes reciprocally activated the platelets, because when we measured the platelet-derived mediators, they were also increased in platelets that were interacting with monocytes compared to platelets infected alone in the ex vivo and also in the in vitro model. So we now know that platelets interact with monocytes, regulating the expression of pro-inflammatory cytokines and monocytes, and this interaction reciprocally amplify platelet activation. So we want to know which adhesion molecules were involved in this interaction. So we performed this interaction, these co-cultures, in the presence of anti-P-selectin antibody, or the abciximab, which is a monoclonal antibody against the integrin alpha-2 beta-3. And we confirmed the participation of both molecules in the induction of tissue factors in monocytes exposed to platelets from severe COVID-19 patients. Both molecules were also involved in the induction of CD16, secretion of TNF-alpha, and secretion of IL-1 beta by the monocyte. So at this point, we know that platelets from severe COVID-19 patients interact with monocytes through P-selectin and integrin alpha-2 beta-3, inducing tissue factor expression, CD16 expression, and the secretion of pro-inflammatory cytokines. But is the adhesion that is inducing all events, or the adhesion induced the tissue factor expression, which is an early event, and then tissue factors amplifies inflammation through the activation of protease-activated receptors? So to answer this question, we also blocked, besides P-selectin and integrin alpha-2 beta-3, we blocked the tissue factor with anti-tissue factor antibody, and we confirmed that the expression of CD16, secretion of TNF-alpha, and secretion of IL-1 beta were all dependent on tissue factor expression. However, many other mediators were induced directly by the adhesion of platelets through P-selectin and integrin alpha-2 beta-3, and were not inhibited by the anti-tissue factor expression. So we confirmed that platelets induce the secretion of many mediators, especially chemokines, and also the expression of tissue factor that amplifies the activation of the monocyte, inducing the pro-inflammatory cytokines, especially TNF-alpha and IL-1 beta. We confirmed these in our in vitro model, which also induced tissue factor expression in monocytes depending on the adhesion of platelets. And in the in vitro model, CD16 expression, TNF-alpha, and IL-1 beta secretion were also dependent on a tissue factor mediated amplification. but many mediators were independent on this tissue factor mediated amplification. And interestingly, we have shown that the platelet-derived mediators were also dependent on tissue factor activity. So we reassure that the platelet-leukocyte interaction reciprocally amplifies platelet activation, and we demonstrate a role for tissue factor in this amplification. We are now interested in, we then investigated the tissue factor receptors involved in this response. So we used PAR1 or a PAR2 selective inhibitor, and we confirmed that the amplification of the monocyte activation was dependent on PAR1 and 2, and the amplification of platelet activation was dependent on PAR1. So we have shown this model in which hypercoagulability and hyperinflammation amplify themselves in COVID-19 depending on platelet monocyte interactions that induce tissue factor, induce inflammatory activation and tissue factor expression in the monocytes, and tissue factor in its turn amplify platelet and monocyte activation, contributing to hypercoagulability and hyperinflammation in COVID-19. All those cellular and molecular events were associated with poorer prognostic in a cohort of ICU-admitted patients. I would like to open a parenthesis here to talk about obesity a little bit, because we receive a lot of questions on how the comorbidities of those patients with COVID-19 are modulating these thromboinflammatory phenomena, because we know that patients with diabetes, obesity, hypertension, and cardiovascular conditions are more likely to evolve to a severe case, and all those comorbidities are already associated with increased thromboinflammation and the higher risk for cardiovascular events. And we observed in our cohort and in many others that obesity in patients with COVID-19 was strongly associated with the risk of severity and the risk of ICU admission when looked separately or in combination with the other comorbidities. So we have recently initiated to investigate those thromboinflammatory phenomena in a cohort of patients with obesity. And of course, we observed that patients with obesity also present increased platelet activation, but it's not like anything like we observe in patients with COVID-19. For example, the expression of P-selectin is not increased in patients with obesity. And the expression of CD63 is mildly increased. In COVID-19, we observe a strong increase in both markers. And not all leukocytes are interacting with platelets during obesity either. In COVID-19, we have observed interaction of platelets with monocytes, neutrophils, B cells, T cells, NK cells. And in obesity, we do not observe increased interaction with T cells, but we observed increased platelet monocyte aggregates formation. And this interaction with monocytes, these interactions of platelets with monocytes during obesity were also associated with CD16 and tissue factor expression in the monocytes. This shows that these events are not exclusive of infection conditions. We believe that platelets interacting with immune cells and regulating innate and adaptive immune response are fundamental phenomena of regulation of the immune response, which is happening in infectious disease, but also in sterile inflammation. So the study with obesity is ongoing, and we have a lot of things ongoing right now. Now that we have achieved higher vaccine coverage, severe COVID-19 looks like a past problem, even though it's not completely eradicated. It's not a health emergency yet it was in 2020, 2021. But we still have to deal with a lot of recovered patients, and we have been aware of this persistence of symptoms in patients that had recovered to severe or from mild COVID-19. And this syndrome has been called post-acute COVID-19 or long COVID. And this syndrome is more often observed in patients that presented viral pneumonia and that has been hospitalized, but it's not exclusive, because people with mild COVID-19 also can evolve to long COVID syndrome. And it's important to note that the vaccination, besides prevent the severe forms of COVID-19, also prevents the persistence of symptoms after recovery. So this is a recent publication in Nature Medicine that showed that COVID-19 survivors, patients that had been COVID-19 in the last year, are at higher risk of develop thromboembolic disorders or cardiovascular outcomes. Of course, the risk is higher in patients that survived the ICU stay or the hospitalization, but the risk is also elevated for patients that have the mild COVID-19 compared to contemporary controls. And the main thromboembolic events were pulmonary embolism and deep vein thrombosis, the same ones that we observed in acute COVID-19. So we have performed a study of COVID-19 survivors. Our group has included 46 COVID-19 survivors. Most of them had moderate COVID-19 pneumonia, but 20% of them had severe cases needing to invasive mechanical ventilation. And we have compared this cohort with 29 healthy donors that were followed since the beginning of the pandemic with weekly nasal swabs to investigate the SARS-CoV-2 infection, and they had never been in contact with SARS-CoV-2 to the moment of inclusion. So we have a full PET-CT scan study of those patients, and those COVID-19 survivors still present evidence of inflammatory infiltration in the lungs, which occurs alongside increased inflammation, evidenced by IL-6 and IL-1 in plasma. Those patients still have increased endothelial activation and vasculopathy, evidenced by markers of endothelial activation, angiopoietins, DCAN, and ICAN. And these patients still have increased hypercoagulability, evidenced by the higher levels of tissue factor and the dimer. The dimer is a product of fibrin degradation, which is a non-direct measure of coagulation activation. So those patients with that survived COVID-19 pneumonia in the last 6 months were still presenting features of systemic inflammation and hypercoagulability. So we asked, is there persistent platelet activation in long COVID-19 patients? We show that both markers of platelet activation, CD62 and CD63, are increased in platelets isolated from these patients. And when put in culture, those platelets from COVID-19 survivors secrete higher levels of platelet-derived chemokines and growth factors. We confirmed higher levels of platelet-derived products in the plasma of these patients. So yes, the answer is platelet activation persisted after at least 6 months after the recovery of severe COVID-19. We also investigated platelet hyperactivity in those patients, and we performed the fibrinogen adhesion assay, and platelets from COVID-19 survivors adhere to a higher area on fibrinogen. And Also, these platelets are hyper-responsive to thrombin. They respond to a suboptimal concentration that's not able to activate platelets from healthy controls, and they respond to a greater extent to optimal thrombin concentration. And finally, we would like to have some idea of which is the trigger of platelet activation, even though even though there is some evidence of viral persistence in COVID-19, these patients are no longer transmitting, and we have no evidence of SARS-CoV-2 in the upper airways. So we tested the hypothesis that some inflammatory coagulation mediator in the plasma of these patients may activate platelets. So we incubated platelets from healthy donors with plasma from another health donor or from a COVID-19 survivor. And the plasma from COVID-19 survivors were able to activate those control platelets. We were able to dissect some of the mechanisms involved in the activation by COVID-19 survivors' plasma. And it was partially inhibited by blocking of the purinergic receptors, integrin alpha-2 beta-3, which is a fibrinogen receptor, and the inhibition of COX-2, suggesting that there is an amplification mediated by fibrinogen, ATP, and thromboxane A2 in the activation induced by plasma, which was inhibited by greater extent with the combination of the three drugs. So we have we have shown that platelet activation persists alongside endothelial activation and systemic inflammation in patients that survived COVID-19 pneumonia, and that the thromboinflammatory milieu of COVID-19 survivors can amplify platelet activation. But we have a lot of open questions. We do not know which is the trigger of platelet activation in this chronic situation, long COVID. And we, among the open questions, we don't know how long it lasts. Remy, our student, which I co-supervise with Patricia Bosa, he is a student of the immunology and inflammation in UFJF, and he is now including the same patients at 18 to 24 months after recovery. And we observed that the platelet activation is reduced, but it is still higher than the median of the control group. So this indicates that this persistent platelet activation can last for at least 2 years in the case of the moderate to severe COVID-19 survivors. But we would also like to know whether this platelet activation also persists in patients recovered from mild COVID-19. So we have been including patients with long COVID that recovered from a mild COVID-19 infection in the last 6 months. And we also observed increased platelet activation and increased platelet responsiveness to thrombin in these long COVID-19 patients. And another open question is whether these thromboinflammatory phenomena contribute to the increased risk of thrombosis on those patients. So that will require longer prospective following of these patients or the study with animal models. And I would like to know from the team of the Laboratory of Immunothrombosis, from the team of the Laboratory of Immunopharmacology, and many other collaborators. So I would like to highlight that the Lab of Immunothrombosis is hiring. We are in need now of students from graduation, post-graduation, or postdocs. So people who would like to go to the interior to leave the rush of the capital and come to Juiz de Fora, just get in contact. Thank you.
Thank you, Eugenio, for a very exciting talk. We usually open for the students first. I don't know if there are any questions. Let me check.
There is one in the chat.
Yeah, from Ana Paula Lepicki. If we can start with this question from Dr. Ana Paula. She says, congratulations, beautiful work. Have you also studied the effect of platelets on epithelial cells in the context of cellular extravasation to the tissue parenchyma?
Yes, that's an interesting question. We have a master student in the lab of Patricia Bosa that is investigating the interaction of platelets with epithelial cells, lung epithelial cells particularly. She had investigated the ability of platelets to modulate these cells regarding the replication of the SARS-CoV-2 and also regarding the secretion of inflammatory mediators. He's about to finish her master degree. If I'm not wrong, platelets did not change the replication phenotype, but modulated the secretion of inflammatory mediators. I don't have a lot of details on the mediators which were regulated by the interaction with platelets, but I'll check with Patricia and Mariana, and feel free to talk with Patricia either. This is a very interesting field.
Great. Actually, I have a question myself. Did you guys check anything on the megakaryocytes side of the story? Are they changed somehow? Do you have any sort of reprogramming due to the infection? Are the platelets that are being made are the same as in a healthy donor?
Yeah, this is a very interesting question, Bruno. We haven't looked at the megakaryocytes in COVID-19, but we had in dengue. During my PhD, I did some experiments in the Guy Zimmerman lab, and I had the opportunity to work with megakaryocytes. And we produced the megakaryocytes from cord blood CD34 positive endothelial cells by culturing them with thrombopoietin and stem cell factors, and then infected to dengue virus when they were mature megakaryocytes. I have data that the pro-platelet production was the same when megakaryocyte was infected or not. There is a lot of papers in the literature that infect the megakaryocyte progenitor and show that they not turn into megakaryocytes, but in my case, it was different because I infected the mature megakaryocytes, and as long as I could see, they produced platelets as well. However, we believe that those platelets are different. We then performed the transcriptomic of infected megakaryocytes alongside the transcriptomic of platelets from patients with dengue. So we have this data published. It's a blood paper. I can send you later. We analyzed that together with the transcriptomic of platelets from patients with influenza H1N1, and we observed a lot of inflammatory genes that were upregulated, including the interferon mediated antiviral response, which was very new in platelets and megakaryocytes at the time. So we have published those. But I believe that those platelets are also different regarding their ability to produce damage. They have the restriction factors mediated by interferon, but those things are not happening alone. We know that cells that are activating antiviral responses are also mediating pro-inflammatory response, as we have have observed in dengue. In COVID-19, we confirmed that platelets also express these restriction factors, but we didn't work with megakaryocytes during the COVID-19 pandemic. But it's an interesting field for future experiments.
Sure, sure. Thank you very much. We also have a question from Miriam Werneck.
Hi, Eugenio. Thank you for your talk.
Hi, Miriam.
Thank you for accepting our invitation. I'm sorry, I might have to leave because I need to teach at 1:30, so I just jump in front of.
Give me 1 second. Let me close some windows because it's raining a lot here, and it's very noisy. Just a minute.
Okay. I think it's gonna rain tomorrow over here, no? Usually it comes from there.
Bruno also have a question, but I don't know how to raise my hand. And there is another question in the chat now.
Much better now, thank you.
Okay, great. Eugenia, it's gonna be a quick question. So you mentioned that the follow-up of the work in collaboration with Patricia, you're looking at the patients now 18 months after the first COVID infection. And I wonder if after the patient has been infected, if the vaccine changes the status of the platelets, or if any other infectious influenza could also have a role in that, and if you have if you have to worry about those same patients having had COVID again, since some of the people who were exposed at first maintained the potential for exposure, so they get COVID several times, and if that could impact that lower decrease on the status of the platelets that you see.
Yes, that's a reviewer question. That's probably what we will have difficult to answer to publish this data. It's very difficult to follow. So the best we can do is interview the patients regarding recent infections, but it's an important confounding factor. Ideally, this I want to have for the mild COVID-19, the long COVID after mild COVID-19 group. It will be important to follow a cohort of patients that fully recovered from COVID-19. This is an ideal control for this situation. So we can have the patients that had COVID-19 and then recovered, and the patients that had COVID-19 and then persisted with the symptoms. So we'll know whether the increased platelet activation and increased inflammation associate with the persistence of the symptoms. And when we go to the follow-up in this situation, the recovered cohort is as exposed to new infections as much as the long COVID cohort. So it will be better controlled this way. For now, we only have the pre-pandemic control group, a naive control group, people that have never been in contact with SARS-CoV-2, but we plan to include this new control group of recovered patients that will be a better control of these conditions than interview the patients regarding the reinfection or new infections. We are now collecting the nasal swab to do a multiplex PCR for SARS-CoV-2, influenza A, influenza B, and syncytial respiratory virus, just to be sure that they are not under an infection in the moment of the blood sampling. But regarding previous infections, it's very hard to control.
Okay. And one quick question. I'm not sure if you had access to the autopsy of severe patients who have died, to look at the brain to see if you have the position, because of thinking about the long COVID symptoms, that a lot of people lose memory, have those neurological symptoms. And I wonder if maybe the severe patients you would have the brain slices and see anything different.
Yeah, this is very interesting, and I'm glad you asked, because I have a collaborator from the FAMED aqui em Juiz de Fora, who is a neuroinflammation researcher, and she is a collaborator in this long COVID study. We have a team of psychologists that perform the cognitive evaluation of those patients, and those with cognitive impairment are subjected to cognitive rehab, and then they will be followed up. And I don't think we have the brain slices from COVID-19 patients. Eliana, which is this collaborator, has some brains from Alzheimer's disease, and we are already buying antibodies to check those platelet leukocyte aggregates in the brains of those patients. But it will be very interesting to have the brains of COVID-19 patients that has been expired, or some patients that presented a thromboembolic complication leading to death during the long COVID syndrome. That would be very interesting. Just have to check how to assess those ethically, since we are following the cohort. And we have also a placenta from a patient that expired from an adverse reaction to the AstraZeneca vaccine, and she was pregnant. So we are interested to label platelets and platelet leukocyte interactions in those tissues either. So this is very interesting. It's a sample difficult to have, but interesting to look at these thromboembolic complications.
Thank you very much, Eugenio.
Thank you, Miriam.
Okay, we actually have two questions in the chat from Mariela Stefani. She says that when you were talking about the platelets aggregates, monocytes and lymphocytes were more cytotoxic, and she was wondering if it would be a more specific response to the infection, or it's in a specific response. And her second point was that you described platelets as an infection platform that leads to thrombosis, and she asks if platelets would also have a role in sterile thrombosis.
Thank you, Mariela, for the question. It's very interesting points. I don't think those platelet responses are exclusive to infections. Our study with obesity also showed increased platelet activation and platelet monocyte interactions in obesity, for example. It's not at the same order of magnitude, because COVID-19 and dengue are acute infections. People are very inflamed at this point of the collection, and obesity. Obesity patients had low-grade inflammation, so we observed that the intensity of platelet activation is also at a lower grade in those patients. But we also observed increased platelet monocyte interactions and regulating the expression of tissue factor, for example, on those monocytes. So these mechanisms may also be involved in the higher risk of thromboembolic events and cardiovascular events in sterile inflammation as obesity. Regarding the platelet lymphocyte interactions inducing a response of cytotoxicity in the lymphocyte, this may be regarding the expression of viral proteins in the platelets, and therefore the cytotoxic T cell response is important to eliminate these infected platelets. But I also don't think it's exclusive for infection. For example, platelet CD8 positive T cell interactions was recently reported to be important to the elimination of platelets during immune thrombocytopenia, which is not an infectious condition, but autoimmune disorder. So I really believe that platelet leukocyte interactions and platelets participating in the regulation of the immune system is a seminal, fundamental process of immune regulation, which is not involved in an exclusive pathology, but in the regulation of the immune system per se.
Perfect. Thank you. We have now a question from Dr. Cristiane Bandeira, and then I will have a question from Lucas Bolini. Cristiane, please.
Hi, Eugenio. Thank you for your talk. It was beautiful, love it, and beautiful historic that you are constructing all these years and your group now. It's beautiful. Congratulations.
Thank you.
I would like to talk about three small things. First, your assays concerning co-cultures of platelets and monocytes, they are beautiful assays. But do you have these two cells isolated, put together, and looking at the interactions between both? Of course, I know that's already very difficult to do that. But in vivo, you would also have a lot of neutrophils. Have you thought about to put the three cellular components together? Do you think that platelets have preference for monocytes than neutrophils? I would expect the other way around. And how neutrophils would interfere with everything that you just showed us.
So, okay, you want to give me more work. Yes. I see now. We started to look to the neutrophils during the pandemic in 2020. We had a huge panel of experiments motivated by the will of do everything, but we were not able to do everything. Despite the will, but neutrophils were one of the most studied cells at that point. So yes. Soon a lot of papers started to pop up on this field, and we gave up on the neutrophils. But we also have unpublished data on platelet neutrophils interactions during severe COVID-19. And a lot of paper came out on platelet neutrophils interactions.
Yeah, I completely understand that, and strategically it was beautiful for you guys. But the thing that I'm asking really, it's how this cell would interfere with your, the crosstalk that you are describing so elegantly.
Okay, say yes. I think that platelets prefer the neutrophils than the monocytes, because when you look to the patient data, we have the percentage of neutrophils that circulate aggregated with platelets are huge, are 60, 70, 80%. And monocytes are 10, 20, 45, 50%. And lymphocytes are 5 to 10.
Got it.
So there is a preference. Neutrophils and platelets like each other a lot. It was curious because in obesity we didn't see platelet neutrophils interactions, but saw platelet monocyte interactions. I thought that was strange at the moment, but when we have the three cells together, well, I think that it would modulate a totally different response, because we are reductionist in our experiment. Yes, so I want to study the monocyte. I look to the monocyte, but if I do the platelet together, the response of the monocyte is totally different. So if I have platelets, monocytes, and endothelial cells, it will be totally different either. Okay. And if I have platelets, monocytes, and neutrophils, so we are reductionist to investigate specific events, but it's important that we keep in mind to have 3D cultures with different cell types working together to better model the immune and inflammatory response, for sure.
Sure. So because of that, and using your word reductionists, you said several times during your presentation that some markers, that you use markers of platelet activation, like PDGF and thromboxane. as only platelets products. I mean, you are comparing them to neutrophils and monocytes when you say that, and you know that eosinophils express and produce both. I mean, I'm going to always talk about eosinophils. So, you know, these molecules, eosinophils can secrete too. They are not exclusive of platelets, okay? And because of that, the third point is, when you are showing the comorbidities. You show beautiful data of obesity, of course, but you have asthma in your graph, and asthma is one that protect from the risks. Did you notice? It's beautiful protection. And I know that this exists already in literature, but you have this thromboinflammation bias that I have never seen before. So it's nice to see the asthma there also protecting. And, you know, platelets also interact with eosinophils. And I was wondering that these guys probably have different counts of eosinophils, probably more elevated eosinophils on circulation. And if eosinophils are part of the mechanism of eosinophils protection, because there are also a lot of works talking about eosinophils as protectors. I mean, eosinophils from Th2 conditions, they are probably protecting against the severity of COVID. So I was wondering if part of the mechanism that I have never seen in literature, it's because neutrophils are interacting with platelets, you know, taking platelets for them and doing other things than with neutrophils and monocytes. I think it would be interesting you think about eosinophils sometime in one point, and I would love to talk with you about it. Okay?
Yes, it would be very great. It's a very interesting hypothesis. Eosinophils are protected, and they also need platelets to support their functions.
And they interact very strongly with platelets. You know, ranches, it's one of the maybe the most strongest agonist of CCR3, that eotaxin receptor, but ranches is stronger at the receptor. So they really talk a lot. There is a very beautiful crosstalk about eosinophils and platelets in several levels. So congratulations again, Eugenio. It was very nice to have you here with us.
Thank you, Cristina.
Thank you. And Lucas Bolini, please.
Hi, Eugenio. Thanks for your talk. I was wondering, because it has been described recently that hormonal replacement therapy for women, and also transgender hormonal replacement therapy for transgender women has been associated with increased thrombosis risk and thrombinflammation risk. And I was wondering whether within your patients, if you checked whether women have an increased risk or have had increased platelet activation, and whether this was associated with estrogen replacement or hormonal replacement, and whether this means that they would have an increased risk for thrombinflammation after COVID-19, or have you checked for that?
Well, good question, Lucas. I didn't check for that, but from the literature, I've seen that the mortality of COVID-19 is higher in men than women, but the risk of thrombosis is higher in women after vaccination. So they removed the AstraZeneca vaccine for women during pregnancy. Okay, no, not all women during pregnancy, because pregnancy per se increases the risk of the thromboembolic events, and that makes a lot of sense, because you talked about hormone therapies for transgender being associated with the thromboembolic events, but the contraceptive hormone therapy in young women is also associated with increased risk for thromboembolic events. So these sexual hormones are strongly associated with thromboembolic events. An interesting data that we have in the long COVID cohort, and here I'm not talking about only my study, but everything we read in the literature, that long COVID is more frequent in women than in men. So estrogen may have a role in this, including a colleague from UFMG has some data regarding that in animal models. So it's about to pop up. It's a very complete and finished, I believe it's already submitted, paper in animal models of long COVID. So for sure, those sexual hormones play a role in the thromboinflammation phenotype. But I didn't look to women in hormone therapy and look at specifically to the risk of that.
And on the other way around, because thinking about the young women that got infected with COVID now, and then they maybe would start hormone replacement therapy in the next years, whether because of menopause or something else. Do you think that COVID infection now would increase the risk for thromboinflammation due to hormone replacement therapy in the future?
Well, probably those things will synergize. When you have something that increases the risk of thromboembolic events, and then you add another thing that increases the risk of thromboembolic events, and then you add a third thing, they will probably synergize to a higher risk.
Thank you.
Thank you.
All right. Thank you very much, Eugenio. It was really a great talk. It was really actually a great way to finish the year for the Immuno webinars. This is the last talk that we are going to have this year. We look forward to meet everybody again early next year when we'll resume the Immuno webinars. And I don't think that we could ask for a better presentation today. And thank you very much again. Don't know if Diego, Cristiano, Miriam, if anybody wants to say anything else.
I just want to wish everybody happy holidays, right? Because we're going to see each other again only in the beginning of next year. It was a great, starting here, it was a great opportunity to meet you all and to join this effort with all of you guys.
Sure. Great. So to everybody, from Diego, myself, Cristiani, Gustavo, Miriam, everybody that's involved with the Immuno webinars, we wish everyone very nice holidays. Enjoy the summer vacations. Thank you, Eugenio, again, and we'll see you next year, 2024. Bye-bye.
Thank you, guys. Thank you for the invitation.
Bye-bye. Merry Christmas.
Thank you,
and Happy New Year.
Eugenio.